In this video, we uncover how a single atomic substitution, the H3K27M mutation, reshapes the epigenetic landscape to drive diffuse midline glioma. We will dissect this pathology in 3 steps. The mechanism, how a mutant histone poisons the PRC2 complex. The consequence, how this unleashes a CREB5-driven oncogenic program. And the therapy, how targeting this specific enhancer vulnerability opens a new treatment avenue. DIPG is an aggressive pediatric brain tumor that arises in the pons, a vital region of the brainstem. Unlike many adult cancers driven by enzyme mutations, this tumor is driven by a mutant histone that alters chromatin regulation, the H3K27M Oncohistone. To understand the crime, we need the normal machinery. PRC2 acts as a writer that places H3K27Me3 marks onto histones. These marks act like molecular stop signs. They compact chromatin and keep developmental genes silent once they are no longer needed. When this machinery is disrupted, the consequences are severe. Loss of these stop signs in key regions of the genome allows proliferative and stem-like programs to persist in the brainstem, promoting tumor growth. Part 2. The mechanism. Lewis et al. 2013 identified the culprit, a lysine to methionine substitution at position 27. In a dominant negative manner, nucleosomes containing H3K27M recruit PRC2 and strongly inhibit its catalytic activity, so the writer can no longer efficiently place stop signs elsewhere in the genome. Justin et al. 2016 revealed the atomic basis for this effect. Zooming into the crystal structure, the mutant methionine side chain occupies the lysine binding pocket. Because methionine is hydrophobic, uncharged, and greasy, it forms tight, sticky contacts within this pocket. Compared with the normal lysine-containing peptide, the H3K27M peptide binds PRC2 with higher affinity and potently inhibits its methyltransferase activity. On the binding curve, the green H3K27M line drops more steeply, meaning PRC2 binds the mutant peptide at much lower concentrations, an example of epigenetic poisoning. The consequence is genome-wide. In Western blots, the H3K27Me3 band is dramatically reduced, and the bar graph shows far fewer tumor nuclei positive for H3K27Me3 compared with H3 wild-type tumors. Poisoning just a fraction of histones is enough to cause a broad collapse in H3K27Me3 levels. Part 3. The consequence. This sets up an epigenetic paradox. The brakes are weakened across the genome, yet not every gene turns on. Instead, specific oncogenic programs become activated. Zhou et al. Zoomed in on the CREB5 locus. Each horizontal track in this figure shows a different chromatin mark or protein across the genome. In normal astrocytes on the left, the dark blue H3K27Me3 track forms a broad peak over CREB5, and the RNA-seq track at the bottom is almost flat. CREB5 is off under a repressive blanket. In H3K27M mutant tumor cells on the right, that blue blanket is gone. Instead, a super-enhancer appears, a wide domain of H3K27 acetylation with strong enhancer peaks. A normal enhancer is like a small on switch. A super enhancer is a cluster of enhancers that acts as a giant volume knob for gene expression. Acetylation recruits BRD4 and other readers that stabilize this active state. Chromatin conformation data show loops connecting the super enhancer to the CREB5 promoter, supporting the idea that physical contact drives CREB5 transcription to oncogenic levels. Why does CREB5 matter? It is a master transcription factor that turns on targets like ID1, which interferes with differentiation and helps lock cells in an aggressive, stem-like state. In neurosphere aspirations, control cells form large, robust spheres, but when CREB5 is knocked down, sphere formation collapses and the tumor loses much of its self-renewal capacity. Part 4. The therapy. So we have found the engine, the CREB5 superenhancer. Can we turn it off? Zhou and colleagues tested ABB075, a BET inhibitor that displaces BRD4, the reader protein required to maintain many superenhancers. Without WERD4, the acetylated enhancer is still there, but the transcriptional machinery can no longer assemble efficiently. To test this, they linked the enhancer to a luciferase reporter, essentially a molecular light bulb. As ABB075 concentration increases, the light signal drops, showing that we can pharmacologically dim this enhancer-driven switch. In mouse models, untreated tumors continue to expand, whereas ABB075-treated tumors grow much more slowly or shrink, demonstrating strong therapeutic potential preclinically. Clinically, radiotherapy is still the standard of care and is mainly palliative. New agents like ONC201 are in trials for H3K27M mutant glioma, targeting stress and metabolic pathways. But ABB075 represents a third wave, precision epigenetic targeting. By attacking the super-enhancer dependency created by H3K27, it hits a core feature of the cancer's wiring. However, BET inhibitors often face adaptive resistance, so future strategies will likely combine them with other agents to prevent tumor escape. Part 5. Conclusion. In summary, we have traced a line from structure to therapy. The crime, a single methionine substitution creates a hydrophobic trap. The weapon, this stalls the PRC2 enzyme, stripping methylation from much of the genome. The consequence, this loss of regulation wakes up the CREB5 superenhancer. The verdict, by targeting this dependency with ABB075, we may be able to hit the tumor at its epigenetic core. Understanding the molecular mechanism linking H3K27M, PRC2 inhibition, and enhancer reprogramming is one of our best hopes for turning this currently incurable disease into a treatable one.